Welcome to Med Together. This is a podcast by the people, for the people, as it were. My name is Chana. I'm a med student in New York. This podcast deals with a lot of med school topics, starting from the basic sciences and building its way up, and hopefully it'll be a bit of review that can help y'all consolidate and synthesize important information and maybe phrase it in a way that's easy to understand. By the way, if you're pre-med or you're a nursing student, or you're just interested in sciencey, medically topics, and you're not in healthcare at all, you're totally welcome to. We don't discriminate. If you're interested, you're cool. Ready to rock? Let's do this. Hey everybody, welcome if you're new, welcome back if you're a repeat listener. Today we're going to talk about heredity, and we're just going to go over some of the fundamentals and try to avoid getting lost in the details anywhere because there's a lot of details and there's a lot of places to get lost. But my intention here is just to kind of hit some of the high yield big points. So, we all get 2 copies of every gene called alleles, one from each parent. And this is called your genotype. The genotype then dictates the phenotype, which is the physical manifestation of the genetics that you inherited. I want to take this opportunity to point out that genetic variation between humans is really not a lot in absolute terms. 99.9% of human DNA is identical, partially because only 2% of our DNA even codes for proteins. Most of the rest has regulatory or other effects that we don't really understand. We used to call it junk DNA. We don't call it that anymore, and we're a little more respectful of it, but we still really have very little idea what most of our DNA does. But we're going to focus on the variations that we do have because that's what makes us different. Mendelian inheritance with purely dominant and purely recessive traits is obviously the most straightforward, and there are some genes that do work that way, but in reality it's almost always more complicated than that. But if we're going to talk straight dominant recessive, what that means is that if the dominant gene is present, it will always be expressed, and the recessive gene will be hidden. The recessive gene is only expressed when there's no dominant gene, and you have to have 2 recessive copies. Most genes that we think about are going to be autosomal, meaning that they're on one of the 22 non-sex chromosomes, but there are also genes carried on the X chromosome, and these are called X-linked genes. In terms of inheritance patterns, here's a general way to think about it, and a way to tell what the inheritance of a given trait is by looking at a pedigree, which is a chart of the disorder within a given family. Pedigrees are drawn in a very specific way, with each generation on the same horizontal line, and children in a direct vertical line from their parents. Males are always depicted as squares, females are depicted as circles. A shaded or filled in shape represents an affected person, and an empty shape will represent an unaffected person. There are a lot more symbols, and it can get very specific and very detailed, but these are kind of the very basic pedigrees that you're going to see. You're going to see squares and circles that may be either filled in or empty. And you have to know what that means. So to go through the sort of basic Mendelian modes of inheritance. First we have autosomal dominant. In this mode of inheritance, males and females are going to be affected equally. It doesn't skip generations. And every affected child has an affected parent. Because remember, this kind of allele, if it's present, there will be disease or the characteristic will be present. So if a child has it, it means they got it from a parent. So every affected child has an affected parent. There is an exception, which we'll go over later, but generally this is true. Now, if we talk about autosomal recessive, this also affects males and females indiscriminately. Affected children generally have healthy parents because usually the case is that each parent is going to be carrying 1 copy of this recessive allele, so they're not going to be affected, but if both of them happen to pass on that allele to the child will be affected. So because of this, the trait appears to skip generations. Now, how would we recognize an X-linked recessive pedigree with a carrier mom? So first of all, if it's an X-linked recessive gene, only males are going to be affected. It's going to appear to skip generations like other recessive genes, and only children of females are going to be affected. That's what you're going to notice. You're going to see like, oh, you had like a bunch of brothers and sisters. Only the sisters had children who were affected. That's a big clue that this might be X-linked. Now, not all of the children are going to be affected. First of all, it's going to be only sons. No daughters are going to be affected, but 50% of the daughters will be carriers, and 50% of the sons will be affected. Because this is, again, if the mom is a carrier. So she has 1 normal X and 1 affected X. So for every child, there's a 50-50 flip which X she's going to give. If she gives the affected X to a male child, that's his only X, so he's going to have the disease. Versus if she gives her affected X to a female child, remember we said it's a recessive gene, the female child has the other healthy X from her father, and she's not going to be affected, but she will be a carrier. Now, What about an X-linked recessive disorder with an affected father? Remember, fathers can't be carriers of X-linked disorders because they only have one X. So if they have the affected X, they have the disease. In this case, all daughters will be carriers because the father has two things to give. He has an X and a Y. If he gives an X, the child is a female, and he only has a diseased X to give. So all of his daughters will be carriers. Now, none of his sons will be affected because he gives all his sons his Y, which are not affected. This is all X-linked recessive, which is much more common. But you can rarely see X-linked dominant traits. So in that case, the mother will be affected because she has two X's. One is affected, one is not, but the affected one is dominant. In that case, again, half of her children will be affected because she still has the one healthy X to give. And since the mom gives X to everyone, it's going to look like any other dominant disease on a pedigree. There's a 50-50 chance of her giving it to a child or not giving it to a child. This is different than X-linked dominant disease with an affected father. If the father is affected, remember he has one X and one Y. So if his X is affected, he's going to give his affected X to all of his daughters. And because this is dominant, all of his daughters will be affected. And again, his sons will not be affected because he's giving his sons his Y, which is perfectly healthy. There are some X-linked dominant traits which are male lethal. So in this case, only affected females will show in the pedigree. And you'll see a high number of male miscarriages. And that's really the thing to look out for, is that affected women are going to have many more female to male children in a 2-to-1 ratio because half of her male children will receive the diseased X and they'll die. Either they'll be stillborn or they'll be miscarried. So you're not going to see any affected males, but you'll see that she has more female children and a high number of male miscarriages. Now there is one other kind of inheritance called mitochondrial inheritance. And this looks dominant because affected women are going to pass it on to all their children. Mitochondrial inheritance is different because all of your mitochondrial DNA comes from your mother. Males don't give over their mitochondrial DNA because if you think about the way that fertilization works, only the egg has mitochondria to give when it's fertilized by the sperm, which has very little going on except for DNA, but not mitochondrial DNA. So all mitochondrial DNA comes from the mother. So in this case, it's going to look like a dominant trait because the affected mother is going to pass it on to all her children. But, and here's where you see that it's mitochondrial and not regular dominant because only the female children are going to pass it on, even though the male children will be affected. So those are Mendelian genetics. Most traits, though, are polygenic, which is to say that they're controlled by more than one gene. There are some disorders like hemophilia, Huntington's, certain types of deafness that are straight inheritance. If you have the mutation, the disease will be present. But it's usually better to think of genes as predictors. They don't always necessitate having a disease. So complex multigenic disorders like diabetes, hypertension have so many causes that genes may put you at risk, but this risk is really modifiable and it's only predictive up to a point. So for everyone, health maintenance is important. It's important that we don't succumb to this so-called genetic determinism. Oh, I have these genes. Oh, my father has diabetes. My mother has hypertension. I'm going to have it because it's genetic. There are genetic factors, certainly, but there are so many modifiable risk factors that have more bearing on it than the genetics, which is your non-modifiable risk factors, that it's important to remind patients of this frequently. Okay, we're going to delve a little bit more into some of the complexities of Mendelian inheritance, because even though we just said the Mendelian inheritance is straightforward, I lied. It's really not. First of all, you have something called variable expressivity, which is the degree to which the phenotype of affected persons is affected by the genotype. You can think of it like a sliding scale of expression among people with the same condition. And the reason why you have variable expressivity is a lot of times because of environment or other modifying genes. And the far end of the expressivity spectrum is penetrance. So penetrance is an all or nothing. Did the genotype affect the phenotype or not? Like, for example, did a person with a BRCA mutation develop cancer? If they didn't, you would say, oh, it's non-penetrant. And variable penetrance can confuse a pedigree, because if a dominant allele is non-penetrant in a person, the trait can appear to skip a generation, and then you'll be like, oh, look, there's a skip generation, it can't be dominant. It can actually be dominant, it could just have variable penetrance. There's another term that's important to know. So, in general, someone who has two of the same copy of an allele, either both dominant or both recessive, is called homozygous, and someone who has one dominant, one recessive is called heterozygous. There is such a thing, however, as a compound heterozygote. Compound heterozygotes have two diseased alleles, but they're different variants, versus homozygotes, which have two copies of the same variant. Another thing that kind of muddles this picture is something called genetic heterogeneity, which is a phenomenon where the same phenotype can be caused by different genotypes. So it's the same disease, but it's caused by different mutations. Now within this, we have two types. There's locus heterogeneity and allelic heterogeneity. So locus heterogeneity is when the alleles at different loci lead to a common phenotype. So I have a mutation in a protein in my ear, which causes deafness, and you have a mutation in a protein in your brain, which causes a neural deafness. So we have the same phenotype, but it's caused by a different genotype, and it's a completely different locus. It's a different protein that's affected, but it leads to a common downstream effect. Versus allelic heterogeneity is when the two different variants are at the same locus, but they're different mutations. The code is supposed to say GCC, my code says GCT, and your code says GCA. So it's the same locus, different variants. We end up with the same looking disease, often because these are, let's say, nonsense mutations or missense mutations, which cause loss of a protein. So again, downstream effect is the same, but it's caused by a different variation. This is important because locus heterogeneity in an autosomal recessive disorder means that sometimes, even if both parents have this recessive disorder, their children could be unaffected because they may have locus heterogeneity, going back to the deafness example. If I have a recessive mutation in a protein in my ear, which causes deafness, and my husband has a recessive mutation in a protein in his brain, which causes deafness, now we're both homozygotes if we have the condition, right? So we're both deaf, but we can have a hearing child because the child will inherit one recessive ear protein from me, but they'll have the dominant, perfectly healthy ear protein from my husband, and vice versa with the brain protein. You can end up with two people who have the same phenotype but different genotypes having healthy children. Another just kind of term to define is pleiotropy, which is when one mutation can have many phenotypic effects. One example of pleiotropy is Marfan syndrome. So this is a human genetic disorder that affects connective tissues. This mutation causes weakness in connective tissues. So this disease is going to have a lot of different effects throughout the body. It commonly affects eyes, heart, blood vessels, skeleton. It's just one mutation, but because connective tissue is such a widespread thing in our bodies, you end up with many phenotypic effects from the same mutation. Another thing, just to muddy the waters further, is clinical heterogeneity. So this is when different phenotypes arise from different variants in the same gene. So let's say the gene that's mutated in achondroplasia, which is a skeletal disorder, can be mutated in other ways, which produce phenotypes that can be either more severe. There's a version of achondroplasia which is perinatal lethal, or you can have a milder version of the disease, hypochondroplasia, depending on where exactly in the gene the mutation occurs. And this can be seen in other diseases as well. Clinical heterogeneity. Different phenotypical manifestation of mutations in the same gene depending on kind of where and what exactly the mutation is. A biallelic autosomal dominant trait. That sounds counterintuitive because we just had said that autosomal dominant traits only need one allele to have the trait, and that is true. But some traits are worse for homozygotes even though they're dominant. So, and I go back to achondroplasia a lot because there's a lot to talk about with achondroplasia, but people who are homozygous for achondroplasia usually have this embryonic lethal version. It's much, much worse versus, let's say, Huntington's, which is another autosomal dominant disease, but it's not affected by being biallelic. If you have one parent with Huntington's or two parents with Huntington's, you end up with the same situation. Genetic anticipation is a disease that gets progressively worse with each generation. What do I mean by that? This usually is seen in diseases with unstable repeat expansions, where you have variable numbers of CPG repeats that lead to neurological disorders like Huntington's. And let's say the first person in the family to have it may have a milder version, but they pass it on to their children and they pass it on to their children. With each generation, it actually gets more severe. We call this genetic anticipation. We also have to talk about mosaicism. And there are two kinds of mosaicism. We have germline mosaicism and somatic. Germline mosaicism is a mosaicism of the sperm or the eggs, where some, let's say a guy has several million sperm, right? So some of his sperm have a mutation and some don't. That's called a germline mutation because it's in the germ cells. And this can cause an autosomal dominant disease in someone with healthy parents, which is very bizarre looking because you don't expect it to pop up. The other kind of mosaicism is somatic mosaicism. This is post-zygotic but still early. So when the the embryo is several cells, maybe 16 cells, maybe, very, very early on, there's a mutation which gives rise to two distinct cell lines within the same person. Because, for example, let's say when the embryo is 4 cells big and one of those 4 cells gets a mutation. So now 1/4 of all the cells in the body are going to have a mutation, but 3/4 will not. It doesn't have to be a disease mutation. It just means that it's different. And actually, all females are mosaics. What do I mean by that? So there was a scientist called Mary Frances Lyon who proposed this theory called random X inactivation. Random X inactivation comes to explain something we call dosage compensation, which is that the phenotypic effect of the X chromosome is the same in the female, which has 2 X chromosomes, as it is in the male, which only has 1 X chromosome. How do we explain that? 1 out of the 2 X chromosomes in females is actually inactivated very early on in embryonic development. And there's a phenomenon called random X inactivation. So basically, very early on, when the embryo is several cells large, 1 X in each cell gets inactivated, and it's random which one is which. So sometimes the mother's X gets inactivated, sometimes the father's X gets inactivated. And this is how women can express an X-linked disorder sometimes, because normally they shouldn't. But if there's uneven X inactivation, so let's say the woman or the female embryo inherited a diseased X from her mother. So normally we would say, oh, it's recessive, you're not going to see it show up. But if in the random X inactivation, she, for whatever reason, inactivated all of her father's X's, now she only has the mother's X. And you lose that protection of the extra X and can actually express the disorder. So when I say that all females are mosaic, what I mean to say is that early on in embryonic development, when these X's are inactivated, some of the mothers are inactivated and some of the fathers are inactivated. So actually, within your cells, you have 2 different populations going on. This is why, for example, female cats can have different colored spots, because the color is carried on the X chromosome, and sometimes they inactivate one and sometimes they inactivate the other, and that's how you end up with calico cats. Fun fact. Okay, we talked about individual genetics. Now we're going to talk a little bit about population genetics. And we're going to use the Hardy-Weinberg law, which is annoying, but it's actually useful, and it takes a little bit of thinking about sometimes, but it's not as complicated as it seems at first. In order to use the Hardy-Weinberg law, there are some basic assumptions we make regarding a population, and not all of them are exactly true, but they're true enough that we're able to use it. These assumptions are that the population is large, mating is random, there are no new mutations, there's no gene flow, which we'll discuss, and that alleles don't affect reproductive fitness, and we'll discuss that also. Now, within this population, let's say we're looking at specific characteristics, and there's a major allele and a minor allele. The major allele is the allele that has the most allele frequency. This is not necessarily the dominant allele, and sometimes if we're looking at multiple things at the same time, it doesn't even have to be more than 50% of the alleles. But we're going to give very binary examples because it's easier to explain that way. So when we define the major allele, we say that it constitutes greater than 50% of all the alleles in the pool, and it's denoted with the letter P. The minor allele is any allele that occurs less frequently than the major allele, and it can never be more than 50%, obviously. And it's denoted by the letter Q. The allele frequency is the percentage of the total alleles in the pool that are major and minor, and the allele frequencies always have to add up to 100%. So let's say you have 75% major alleles, you have 25% minor alleles. So the Hardy-Weinberg law is made up of 2 mathematical expressions, and we use them as a system of equations. So the Hardy-Weinberg equations state that P plus Q equals 1, which is what we just said. The allele frequencies have to add up to 100%, and that P squared plus 2PQ plus Q squared equals 1. Well, what does this second one mean? This second equation is describing genotype frequencies. So it's relating the probability of drawing 2 major alleles in a row, making someone homozygous major, and with the probability of drawing 2 minor alleles in a row, homozygous minor, and the probability of drawing 1 of each. Remember, when we talk about probability of 2 independent events occurring, we multiply the probabilities. So P squared represents the probability that you will get 2 P alleles. If you're drawing from this random pool, imagine like a big pool of alleles, and you close your eyes and you stick your hand in. The probability of you drawing 2 P alleles in a row, making a homozygote P person, is P squared. The probability of you putting your hand in and drawing two Q alleles in a row, making a Q homozygous person, is Q squared. And then the reason why the middle term for heterozygotes is 2PQ is because it's saying you can do PQ in two different ways. You could stick your hand in and draw a P and then a Q, or you could stick your hand in and draw a Q and then a P. And so we add those two together, we call it 2PQ. And altogether, these have to add up to 1 because you've accounted for all possible genotypes. So P squared plus 2PQ plus Q squared equals 1. Those are all the possible genotypes and all the possible ways to get them. And we can use this, if you know either P or Q, we can use it to find the other one, and we can use this to find the probability that someone will be a heterozygote or that somebody will be homozygous major or homozygous minor, depending on the allele frequency in the genetic pool. Given any one of the variables, we have this two-equation system which can be used to calculate any of the other variables, whatever we're looking for. Now, how do X-linked conditions fit into Hardy-Weinberg? For women, it works exactly like the regular calculation of a recessive disorder. They would need to be homozygous to be affected, and otherwise they're a carrier. For males, you can actually just use P plus Q equals 1, because there is no two events. Males only have one X, so their genotype frequency is the same as the allele frequency of the disease allele. Now, in theory, according to Hardy-Weinberg, there should be constant allele frequencies between generations, but selection, new mutations, genetic drift, and genetic flow all can mean that we don't actually have exactly constant allele frequencies between generations, and we'll talk about what these are a little bit later. Before that, I want to talk about genetic fitness. What is genetic fitness? This concerns the ability of genes to be passed on, and it's denoted by the letter F, F as in Frank. Genetic lethal alleles, by the way, are not necessarily lethal to the person carrying them, although they can be. Often they are, but what it means is that they're lethal to the gene. The gene can't be passed on, either because it causes sterility or death before reproductive age. And these alleles have a fitness of F equals 0. It never gets passed on. If a mutation does not affect gene transmission, like Huntington's, because Huntington's develops later in life in 40s or 50s, after someone has already had children, F equals 1. Most mutations are going to be somewhere on the spectrum. Anything, obviously, that is embryonic lethal or perinatal lethal is going to have a fitness of 0. Things like Huntington's that have a fitness of 1 are pretty rare. And then we have this important concept of how genetic fitness is related to the rate of de novo mutations. They're inversely related. The greater the fitness, the fewer new mutations there will be, percentage-wise, and vice versa. For example, in a disease like thanatophoric dysplasia, which is a severe skeletal disorder characterized by short limbs, folds of extra skin, and is perinatal lethal, the fitness rate is 0. So all mutations must be de novo mutations because these children will never reproduce. Contrast that with Huntington's, which has a later onset, doesn't affect reproductive ability, so genetic fitness is 1 or close to 1, and de novo mutation rate is close to 0. It's also important to make a distinction between de novo autosomal recessive mutations and de novo autosomal dominant mutations. De novo autosomal recessive mutations have very little effect on population phenotypes because the likelihood of someone with a de novo recessive mutation also having inherited a copy of the mutation to make them homozygous, or having 2 de novo mutations, is vanishingly small. So most of the alleles with de novo recessive mutations are going to be held in heterozygotes. So therefore, they're going to be less at risk to be selected against if they're genetic lethal, meaning that genetic lethal diseases are more commonly recessive. Because, for example, a disease like Tay-Sachs, which is a terrible, terrible disease, causes death at a young age, it's a recessive disease, and it's fairly common because people who are carriers are not affected, and then you can pass it on. So most of the Tay-Sachs alleles floating around are held in heterozygotes and then passed on, and they're not selected against in the same way that autosomal dominant are. Autosomal dominant new mutations don't have the buffer of heterozygotes to be able to pass them on, so if they're genetic lethal, they're going to be automatically selected against, and they can only be established through continued new mutations. Now, the coefficient of selection, which is the additive inverse of fitness, tells what proportion of mutant alleles will not be passed on. So S, which is the coefficient of selection, plus F is going to equal 1. For example, something like Huntington's. F equals 1, so S is going to equal 0, which means that all mutant alleles will be passed on. Now, going back to our other example of thanatophoric dysplasia, F equals 0, so S equals 1, which means that all of the mutant alleles will not be passed on. This is just another way of expressing this proportion of how many of these disorders are inherited versus how many are de novo mutations. And I'm going to say this until I'm blue in the face, the more it affects the person early on in life, either making them dead or sterile, the less likely it is that it's going to get passed on, and the greater percentage of this disease will have to be from de novo mutations because it can't be passed on. Now, to talk about some things that affect Hardy-Weinberg. First of all, we have something called assortative mating. This is anything other than random mating. It can be positive assortative mating, where people seek mates with similar traits, or it can be negative assortative mating, where we seek mates with deliberately different traits. And that's something we actually probably do more often with now carrier screenings and things like that. If I know that I'm a carrier of a Tay-Sachs gene, I'm going to deliberately only seek out people who do not carry a Tay-Sachs gene, because that means my children won't be affected. Another thing that can affect population genetics is stratification, which is mating within a social subgroup. And this can interestingly cause a genetic subgroup where certain alleles have a much higher frequency than in the general population. If blue-eyed people only marry blue-eyed people, you're going to have a really bizarre amount of blue-eyed children, which you normally wouldn't expect. Now, blue eyes is not a social subgroup, but I don't want to get into social topics, so I'm going to leave it with blue eyes and let you extrapolate further. Another thing that can affect population genetics, which is similar, is consanguinity, which means marrying a blood relative. So similar to stratification, mating among blood relatives increases the effective frequency of recessive alleles and causes increased risk of autosomal recessive disorders. Genetic drift is a change in allele frequency that is unrelated to the genetic fitness of the allele. Examples of genetic drift are the founder effect and the bottleneck effect, where a small population isolates and creates a genetic subgroup. By chance, the portion of the gene pool in that small group can contain a higher proportion of one or more particular disease alleles, creating a subset of the population with a different allele frequency than the general population. This founder effect is something that we see in certain communities, like, for example, the Ashkenazi Jewish community. Things like BRCA mutations causing breast cancer, Tay-Sachs disease, other recessive diseases are very, very common in this community because they have this founder effect where it's a relatively small population that self-isolates and only or mostly marries within itself, and you end up with this cultivating a genetic pool that's very different from the general population. One last thing to talk about is gene flow. This is changes to population allele frequency because of migration of large groups of people, which will create a mixture of what used to be separate gene pools and change allele frequencies. While this is more common as the world becomes more connected and people don't just marry someone from the next village, it doesn't happen between one generation and the next, which is why it doesn't really change the way we can practically use Hardy-Weinberg. So even though we do have gene flow, we ignore it and we pretend that we don't. And Hardy-Weinberg is still actually pretty effective when we want to use it within our population. Quick wrap up now. We've gone over the basics of Mendelian heredity, diseases that are dominant or recessive, their expected patterns. We touched on non-Mendelian multifactorial disease, and we talked about population genetics using the Hardy-Weinberg law. So that's it for now. Thank you so much for listening. Please reach out to me by email, medtogether26@gmail.com with comments, questions, requests. I would love to hear from you.